Close your eyes and imagine the Earth as it was 4.5 billion years ago. The planet was young, restless and volcanic, a turbulent sphere of molten rock spinning through the cosmic void. Meteors crashed into its surface like celestial raindrops, each impact releasing tremendous energy that kept the surface in a constant state of flux. The atmosphere was thick with methane, ammonia, water vapor, and other gases that would be toxic to most life today. There was no oxygen to breathe, no green plants to see, no birdsong to hear, only the rumbling of the restless Earth itself, and the occasional thunder of cosmic impacts. Yet beneath this seemingly hostile exterior, something extraordinary was beginning to stir. As the planet slowly cooled, water vapor condensed into the first oceans, vast warm seas that would become the cradle of all life. These ancient waters were different from today's oceans, rich with dissolved minerals, organic compounds, and the chemical building blocks of life itself. In these warm, shallow pools of primordial soup, complex molecules were dancing together in ways they had never danced before. Carbon atoms, those versatile architects of life, linked hands with hydrogen, oxygen, nitrogen and sulfur, forming the first organic compounds. These weren't alive yet. They lacked the organization, the purpose, the spark that defines life, but they were the essential building blocks. Amino acids that would one day form proteins, nucleotides that would become DNA and RNA, and simple sugars that would fuel the first metabolisms. Picture these molecules as tiny dancers in an endless ballroom. The ancient seas themselves, they swirled and combined in countless ways, driven by the energy of lightning strikes, volcanic activity, and the gentle warmth of a younger sun. Most of these molecular combinations were fleeting, dissolving back into the warm waters like sugar in tea. But occasionally, just occasionally, something special happened. A combination that was stable, that persisted, that had properties its individual components lacked. The Miller-Urey experiment, conducted in 1953, gave us a glimpse of what these ancient seas might have been like. When Stanley Miller and Harold Urey recreated the conditions of early Earth in their laboratory, mixing water, methane, ammonia and hydrogen in a sealed flask and subjecting the mixture to electrical sparks, they discovered that amino acids, the building blocks of proteins, formed spontaneously. This simple experiment showed that the basic chemistry of life could arise naturally from non-living matter, given the right conditions. But the formation of amino acids was just the beginning. The real mystery was how these simple organic compounds could organize themselves into something truly alive. The answer lay in the remarkable properties of a molecule that can still be found in every living cell today, RNA. RNA, that humble cousin of DNA, was likely one of the first self-replicating molecules. Unlike DNA, which needs a complex cellular machinery to copy itself, RNA can act as both a carrier of genetic information and a catalyst for chemical reactions. Imagine RNA strands floating in the primordial soup, slowly learning to copy themselves through a process of trial and error that would take millions of years to perfect. These early RNA molecules were like the first storytellers in the history of life, passing on their molecular tales to their copies. But they were imperfect storytellers. They made mistakes, introducing small changes with each copying cycle. Most of these changes were harmful, causing the RNA to lose its ability to replicate. But occasionally a change would improve the RNA's ability to copy itself or to survive in its environment. These improved versions would become more common over time, while the less successful versions would gradually disappear. This was natural selection in its most primitive form, not yet operating on living organisms, but on simple molecules competing for resources in the primordial soup. The RNA molecules that were better at copying themselves, or that could survive longer in harsh conditions, left more copies of themselves for future generations. Over millions of years, this process led to RNA molecules of increasing complexity and sophistication. Some RNA molecules developed the ability to catalyze their own formation, speeding up the replication process. Others learned to incorporate amino acids into their structure, creating the first primitive proteins. Still others discovered how to build protective shells around themselves, the precursors to the first cell membranes. As these RNA-based systems became more complex, they began to take on the characteristics we associate with life. They could reproduce, they could evolve, and they could respond to their environment. They were still incredibly simple by modern standards, but they represented a crucial threshold, the transition from non-living chemistry to living biology, the birth of the first cells. As millions of years passed like slow, peaceful breaths, these self-replicating RNA systems began to gather together in small clusters. They found that cooperation offered advantages that individual molecules couldn't achieve. Some RNA molecules specialized in replication, others in metabolism, and still others in protection from environmental hazards. These early molecular communities eventually wrapped themselves in protective membranes made of simple fats called phospholipids. These membranes were permeable enough to allow nutrients to enter and waste products to exit, but stable enough to maintain the internal environment necessary for the complex chemistry of life. These were the first cells, microscopic bubbles of life floating in the ancient oceans like tiny planets in a vast liquid universe. These early cells, called prokaryotes, were beautifully simple in their organization. They had no nucleus, no mitochondria, no chloroplasts, none of the complex internal structures that characterize the cells of more advanced organisms. Their genetic material floated freely in their watery interior, a single circular chromosome that contained all the information necessary for life. Despite their simplicity, these early prokaryotes were remarkably successful. They were the bacteria and archaea that would dominate Earth for the next 2 billion years, diversifying into countless forms and learning to exploit every available niche in the ancient world. Some became scavengers, breaking down dead organic matter and recycling its components. Others became parasites, living inside or on other organisms. Still others became predators, actively hunting and consuming their neighbors. But perhaps the most important innovation of these early cells was the development of photosynthesis. Some bacteria discovered that they could harvest energy directly from sunlight, using it to convert carbon dioxide and water into organic compounds. This was a revolutionary development that would transform not just the living world, but the entire planet. The first photosynthetic bacteria were quite different from modern plants. They used hydrogen sulfide or other compounds as electron donors rather than water, and they didn't produce oxygen as a byproduct. But eventually a group of bacteria called cyanobacteria evolved a more sophisticated form of photosynthesis that could split water molecules and release oxygen gas. This innovation had profound consequences for the early Earth. For the first time in the planet's history, oxygen began to accumulate in the atmosphere. This wasn't necessarily a good thing for most of the organisms alive at the time. Oxygen was actually toxic to many of them, causing a mass extinction event known as the Great Oxidation Event, or the oxygen catastrophe. Picture this slow transformation of the ancient world. For nearly 1 billion years, oxygen levels gradually rose, turning the skies from their original orange-brown color to the blue we know today. The iron dissolved in the oceans reacted with the oxygen to form rust, which settled to the bottom of the sea in distinctive red layers that geologists call banded iron formations. These formations serve as a record of the Earth's transition from an oxygen-free world to an oxygen-rich one. The Great Oxidation Event was like the Earth learning to breathe for the first time. It was a gradual process, as gentle and inevitable as the rising sun, but it fundamentally changed the nature of life on our planet. Those organisms that couldn't adapt to the new oxygen-rich environment perished, while those that could learn to use oxygen for their own metabolic processes, gaining access to far more energy than was available through fermentation alone. This transition also led to the formation of the ozone layer, a protective shield of oxygen molecules high in the atmosphere that would block harmful ultraviolet radiation from reaching the Earth's surface. With this protection in place, life could eventually venture out of the protective depths of the oceans and colonize the land. The emergence of complex cells about 2 billion years ago in the warm, oxygen-rich seas of the Middle Proterozoic era, something extraordinary happened that would change the course of evolution forever. A larger bacterial cell engulfed a smaller one, but instead of digesting its prey, the two organisms began to live together in a partnership that would benefit both. This event, known as endosymbiosis, was like the first diplomatic treaty in the microscopic world. The larger cell, called the host, provided protection, nutrients, and a stable environment for the smaller cell to live in. In return, the smaller cell, now called an endosymbiont, provided specialized services that the host cell couldn't perform on its own. The most important of these endosymbiotic relationships involved aerobic bacteria, organisms that had learned to use oxygen for respiration. When these bacteria were engulfed by larger cells, they continued to live and reproduce inside their hosts, providing them with a steady supply of energy in the form of ATP, the universal energy currency of life. Over millions of years, These endosymbiotic bacteria became so specialized for their role that they could no longer survive independently. They became organelles called mitochondria, the powerhouses of the cell that still reside in every eukaryotic cell today. The mitochondria in your own cells are the direct descendants of these ancient bacterial partners, quietly working to power your every thought and movement. Some eukaryotic cells also engulfed photosynthetic bacteria, which became chloroplasts, the green organelles that allow plants to capture sunlight and convert it into chemical energy. These chloroplasts retained their own DNA and their own ribosomes, evidence of their ancient bacterial origins. The evolution of eukaryotic cells was a major transition in the history of life. These new cells were larger and more complex than their prokaryotic ancestors, with specialized internal compartments that allowed for more sophisticated biochemistry. They had a true nucleus, bounded by a membrane, where the genetic material was stored and protected. They developed an internal skeleton made of protein fibers that gave them shape and allowed them to move in more complex ways. But perhaps most importantly, eukaryotic cells developed the ability to reproduce sexually. Instead of simply dividing to create identical copies of themselves, they could exchange genetic material with other cells, creating offspring that were genetically unique. The revolutionary dance of sexual reproduction. The evolution of sexual reproduction was one of the most important innovations in the history of life. Though it initially seems puzzling from an evolutionary perspective, why would an organism give up the ability to pass on 100% of its genes to its offspring, settling instead for just 50%? Why complicate the relatively simple process of asexual reproduction with the need to find a mate? The answer lies in the tremendous advantages that sexual reproduction provides. When two organisms mate, they combine their genetic material in new ways, creating offspring that carry novel combinations of traits from both parents. This genetic recombination is like shuffling a deck of cards. It creates new combinations that have never existed before. Imagine the first eukaryotic cells tentatively reaching out to each other across the microscopic distances of their ancient world. Some cells developed specialized structures that allowed them to recognize and fuse with compatible partners. Others evolved the ability to produce gametes, specialized reproductive cells that carried only half of the parent's genetic material. This genetic shuffling created tremendous raw material for natural selection to work with. In an asexually reproducing population, all individuals are genetically identical clones. If the environment changes, the entire population might lack the genetic variation necessary to survive. But in a sexually reproducing population, the constant recombination of genetic material ensures that each generation contains individuals with slightly different combinations of traits. Sexual reproduction also provides a powerful defense against parasites and pathogens. These harmful organisms often evolve to exploit specific weaknesses in their hosts. In an asexually reproducing population, once a parasite learns to exploit a particular genetic weakness, it can devastate the entire population. But in a sexually reproducing population, the constant shuffling of genetic material means that parasites must constantly adapt to new host genotypes. The evolution of sexual reproduction also led to the development of complex life cycles. Many organisms began to alternate between haploid phases with one copy of each chromosome and diploid phases with two copies of each chromosome. This alternation allowed for even more genetic variation and provided additional opportunities for natural selection to act. As sexual reproduction became more sophisticated, organisms evolved increasingly elaborate mechanisms for finding mates and ensuring successful reproduction. Some developed chemical signals that could attract partners from great distances. Others evolved complex courtship behaviors or competitive displays. Still others developed specialized reproductive organs that could deliver genetic material more efficiently. The explosion of multicellular life. For more than 1 billion years after the evolution of eukaryotic cells, life remained largely microscopic. Single-celled organisms dominated the oceans, gradually becoming more complex and diverse, but still operating as individual units. Then, about 600 million years ago, something remarkable began to happen. Cells started working together in permanent partnerships, giving rise to the first multicellular organisms. The transition to multicellularity was like the formation of the first cities in human history. Just as people discovered that they could accomplish more by working together than by living alone, cells found that cooperation offered advantages that no individual cell could achieve. Some cells specialized in obtaining nutrients, others in reproduction, and still others in protection or movement. The earliest multicellular organisms were simple colonial forms, groups of identical cells that lived together but maintained their individual identities. These colonies were like communes where everyone contributed to the common good but retained their independence. Over time, however, the cells in these colonies began to specialize, taking on different roles and losing their ability to survive on their own. This specialization led to the evolution of tissues, groups of cells that worked together to perform specific functions. Muscle tissue could contract to produce movement. Nerve tissue could transmit electrical signals, and epithelial tissue could form protective barriers. The development of tissues was followed by the evolution of organs, complex structures made of multiple tissue types that could perform sophisticated functions. The advantages of multicellularity were enormous. Multicellular organisms could grow larger than single cells, allowing them to exploit new ecological niches and avoid predation by smaller organisms. They could develop specialized sensory organs that could detect food, mates, or danger from great distances. They could evolve complex behaviors that would be impossible for single cells. But multicellularity also presented new challenges. How could cells in different parts of the organism communicate with each other? How could they coordinate their activities to work toward common goals? How could they ensure that selfish cells didn't exploit the system for their own benefit? The solutions to these problems led to the evolution of some of the most sophisticated biological systems we know. Nervous systems evolved to coordinate behavior and respond to environmental changes. Circulatory systems developed to transport nutrients and remove waste products. Immune systems arose to protect the organism from disease and to eliminate rogue cells that might threaten the whole. The Cambrian explosion, life's greatest awakening for billions of years, life had remained largely microscopic and simple. The fossil record from this vast stretch of time, known as the Precambrian, contains mostly bacterial mats, simple algae, and a few enigmatic soft-bodied creatures. Then, about 540 million years ago, something extraordinary happened that would forever change the face of life on Earth. In a relatively short period of geological time, perhaps as little as 10 million years, life exploded into an incredible diversity of forms. This event, known as the Cambrian explosion, marked the beginning of the Phanerozoic Eon, the age of visible life. Suddenly, the fossil record teemed with creatures that seemed to come from another world entirely. The Cambrian seas were filled with trilobites, arthropods with compound eyes that could detect polarized light, and sea underwater with remarkable clarity. There were anomalocarids, massive predators with grasping arms that could reach 2 meters in length. Hallucigenia walked on 7 pairs of spiny legs, its back covered with defensive spikes. Opabinia had 5 eyes and a bizarre proboscis tipped with a grasping claw. These weren't just bacteria anymore. They were complex animals with sophisticated organ systems, behaviors, and ecological relationships. They had eyes that could form images, brains that could process information, and appendages that could manipulate their environment. Many had hard shells or skeletons that could preserve in the fossil record, giving us our first clear window into the diversity of ancient life. The key innovation that made the Cambrian explosion possible was the evolution of complex developmental programs. Through a process called gene duplication and divergence, organisms acquired multiple copies of important developmental genes. These extra copies were free to evolve new functions without disrupting the essential processes needed for survival. One particularly important group of genes, called the Hox genes, evolved to control the development of body segments and appendages. These genes act like molecular architects, determining where legs, eyes, and other structures will form during embryonic development. The duplication and diversification of Hox genes allowed for the evolution of increasingly complex body plans. The Cambrian explosion also coincided with important changes in the ocean environment. Oxygen levels had reached a threshold that could support larger, more active organisms. The development of hard shells and skeletons was made possible by the availability of calcium carbonate in the oceans. Predation pressure increased, driving the evolution of both better defensive structures and more sophisticated hunting strategies. During this remarkable period, most of the major animal phyla that exist today first appeared in the fossil record, the ancestors of vertebrates, arthropods, mollusks, echinoderms, and cnidarians, all emerged from the Cambrian seas. It was as if life was conducting a vast experiment, testing every possible form and function to see what would work in the new, complex ecosystems that were emerging. The Cambrian explosion was not just about the evolution of individual organisms, it was about the evolution of entire ecosystems. For the first time in Earth's history, complex food webs emerged, with primary producers at the base, herbivores in the middle, and predators at the top. The interactions between these organisms drove further evolution, creating feedback loops that accelerated the pace of change. The great venture, life's conquest of land for hundreds of millions of years after the Cambrian explosion, life remained largely confined to the oceans and other aquatic environments. The land was a harsh, forbidding place, dry, exposed to intense ultraviolet radiation, and lacking the nutrients and support that water provided. Yet gradually, some organisms began to venture into this hostile realm, setting the stage for one of the most dramatic transitions in the history of life. The first pioneers were likely simple plants and fungi that formed partnerships near the water's edge. These early partnerships, called mycorrhizae, were mutually beneficial relationships where fungi helped plants absorb nutrients from the soil, while plants provided the fungi with sugars produced through photosynthesis. Together, they could survive in environments that neither could tolerate alone. These early land plants were nothing like the towering trees and colourful flowers we see today. They were small, simple organisms that hugged the ground and reproduced through spores rather than seeds. They had no true roots, stems or leaves, but they began the slow process of transforming the terrestrial environment. Their remains contributed to the formation of the first soils. Their photosynthesis continued to add oxygen to the atmosphere, and their presence provided food and shelter for the animals that would eventually follow. The evolution of vascular tissue, specialized cells that could transport water and nutrients throughout the plant body, was a crucial innovation that allowed plants to grow larger and venture farther from water sources. These early vascular plants could develop root systems that could tap into soil moisture and nutrients, and they could grow tall enough to compete for sunlight. As plants established themselves on land, they began to modify the environment in ways that made it more hospitable for other forms of life. They created shade that reduced temperature extremes. Their root systems helped prevent soil erosion, and their tissues provided organic matter that enriched the soil. They also began to produce oxygen through photosynthesis, further increasing the oxygen content of the atmosphere. The transition of animals from water to land was even more challenging than the plant transition. Water provides support against gravity, maintains body temperature, and facilitates the exchange of gases and nutrients. Animals venturing onto land needed to develop new solutions to all of these problems. The first animals to make the transition were likely arthropods, creatures with jointed legs and external skeletons that could provide support in the absence of water. These early terrestrial arthropods were probably predators and scavengers that ventured onto land to hunt for food and returned to water to reproduce. Fish in the shallow waters of Lake Devonian seas began to face increasing competition and predation pressure. Some species developed stronger fins and primitive lungs that allowed them to gulp air from the surface. These adaptations were initially useful for surviving in oxygen-poor waters, but they also pre-adapted these fish for life on land. Over millions of years, the fins of these fish became more robust and leg-like, allowing them to support their weight on land for increasingly long periods. Their gills became less efficient, but their lungs became more sophisticated. Their lateral line system, which detects water movement, became less important, while their other senses adapted to function in air. Picture these pioneering creatures, the first amphibians, awkwardly pulling themselves onto muddy shores with their still-webbed feet. Their bodies were still more suited to water than land. Their skin was permeable and prone to drying out, and they had to return to water to reproduce. But they were driven by some ancient instinct to explore new frontiers, to exploit the abundant resources that the land provided. These early amphibians faced numerous challenges on land. They had to develop new ways of breathing, since gills don't function in air. They had to evolve stronger skeletons to support their bodies against gravity. They had to develop new sensory systems to navigate in a world where sound, smell, and vision worked differently than in water. But the rewards were substantial. The land offered new food sources, new places to live, and escape from the intense predation pressure of the oceans. Those amphibians that could spend more time on land had access to resources that were unavailable to their purely aquatic relatives. The rise of the vertebrates as life established itself on land vertebrates began to diversify in remarkable ways. The early amphibians gave rise to a spectacular array of forms, from small salamander-like creatures to massive crocodile-like predators. But amphibians were still tied to water for reproduction. Their eggs lacked protective shells and would dry out if laid on land. The evolution of the amniotic egg was a revolutionary development that freed vertebrates from their dependence on water. This innovation involved the development of several new membrane structures that could protect and nourish the developing embryo. The amnion formed a fluid-filled sac that provided the embryo with its own private aquatic environment. The allantois served as a waste disposal system, while the chorion facilitated gas exchange with the outside world. The creatures that first developed amniotic eggs were the ancestors of all modern reptiles, birds, and mammals. These early amniotes could lay their eggs on land, opening up vast new territories for colonization. They also developed more efficient kidneys that could conserve water, and many evolved waterproof skin that reduced moisture loss. The early reptiles were small, lizard-like creatures that lived primarily on insects and other small prey. But as they adapted to terrestrial life, they began to diversify into an incredible variety of forms. Some became large herbivores that could process tough plant material. Others became swift predators that could chase down prey across open landscapes. Still others developed the ability to climb trees or even glide through the air. The age of dinosaurs, which lasted from about 230 to 65 million years ago, was a time of incredible experimentation in vertebrate design. Dinosaurs evolved from small bipedal archosaurs and rapidly diversified into forms that filled nearly every terrestrial niche. Some dinosaurs, like the massive sauropods, were enormous herbivores with long necks that could reach vegetation high in the trees. Others, like Tyrannosaurus rex, were apex predators with powerful jaws and keen senses. But perhaps the most remarkable dinosaur innovation was the evolution of flight. Some small bipedal dinosaurs developed feathers, initially probably for insulation or display. Over time, these feathers became more sophisticated, eventually allowing for powered flight. These flying dinosaurs were the ancestors of modern birds, and they represent one of the most successful transitions in the history of life. During the Mesozoic era, while dinosaurs dominated the land, small furry creatures scurried through the underbrush, avoiding the attention of their giant reptilian contemporaries. These were the early mammals, descendants of cynodont therapsids that had evolved several crucial innovations. They were warm-blooded, which allowed them to remain active in cooler temperatures. They nourished their young with milk, providing excellent nutrition during the critical early stages of life, and they had larger brains relative to their body size, which allowed for more complex behaviors and better problem-solving abilities. These early mammals were mostly small, nocturnal creatures that fed on insects, seeds, and other small food items. They had to remain inconspicuous to avoid predation by dinosaurs, but they were developing the physiological and behavioral innovations that would later allow them to dominate terrestrial ecosystems. The Great Extinction and Renewal 65 million years ago, as the Cretaceous period was drawing to a close, Earth was struck by a cosmic catastrophe that would forever change the trajectory of life. A massive asteroid, perhaps 10 kilometers in diameter, slammed into the Earth near what is now the Yucatan Peninsula in Mexico. The impact was so powerful that it released energy equivalent to billions of nuclear bombs, sending shockwaves around the globe and throwing massive amounts of dust and debris into the atmosphere. The immediate effects of the impact were devastating. The collision vaporized the asteroid and a large portion of the Earth's crust, creating a fireball that ignited forests across much of North America. The debris thrown into the atmosphere blocked out the sun for months, causing global temperatures to plummet and photosynthesis to virtually cease. Acid rain fell from the darkened skies, poisoning lakes and rivers. This cosmic catastrophe, known as the Cretaceous-Paleogene extinction event, ended the age of dinosaurs and eliminated roughly 75% of all species on Earth. The massive sauropods, the fearsome predators, and the diverse array of smaller dinosaurs all vanished from the fossil record. Along with them disappeared many marine reptiles, flying reptiles, and countless other forms of life. But extinction events, while tragic, also create opportunities for innovation and renewal. The mammals, small and adaptable, survived the extinction event and found themselves in a world suddenly free of their giant competitors. Ecological niches that had been occupied by dinosaurs for 165 million years were suddenly empty, creating unprecedented opportunities for diversification. The early Paleogene period, immediately following the extinction, was a time of rapid evolutionary change. mammals began to diversify at an extraordinary rate, evolving into forms that filled the ecological roles previously occupied by dinosaurs. Some became large herbivores, developing complex digestive systems that could process tough plant material. Others became swift predators, evolving keen senses and sophisticated hunting strategies. Still others took to the trees, developing grasping hands and stereoscopic vision. This adaptive radiation of mammals was one of the most dramatic evolutionary events in the history of life. Within just a few million years, mammals had evolved from small, shrew-like creatures into a diverse array of forms that included the ancestors of horses, whales, bats, and primates. The speed of this diversification shows how quickly evolution can operate when new ecological opportunities become available. The extinction of the dinosaurs also had profound effects on plant communities. Many flowering plants had co-evolved with dinosaurs, relying on them for seed dispersal and other services. The loss of these large herbivores led to changes in plant communities and opened up new opportunities for the evolution of different plant forms. This pattern of extinction and renewal has repeated throughout Earth's history. There have been 5 major mass extinctions, each followed by periods of rapid diversification and the evolution of new forms of life. The Permian-Triassic extinction, 252 million years ago, eliminated 96% of marine species and 70% of terrestrial species. The late Ordovician extinction, 445 million years ago, eliminated 85% of marine species. Each of these events was followed by evolutionary innovations that led to new forms of complexity and diversity. The evolution of intelligence and social behavior among the mammals that diversified after the extinction of the dinosaurs were the primates, a group that would eventually give rise to humans. The early primates were small, arboreal creatures that lived in the tropical forests of the Paleocene and Eocene epochs. They were characterized by several key innovations that would prove crucial for the later evolution of intelligence. Living in the three-dimensional world of the forest canopy required sophisticated spatial reasoning and hand-eye coordination. Primates evolved grasping hands with opposable thumbs that could manipulate objects with precision. They developed stereoscopic vision that allowed them to judge distances accurately when leaping from branch to branch. Their brains enlarged to process the complex sensory information needed for arboreal life. Perhaps most importantly, primates evolved complex social behaviors. Living in groups provided protection from predators and allowed for cooperative foraging and child rearing. But group life also required the ability to recognize individuals, remember past interactions, and navigate complex social hierarchies. These cognitive demands drove the evolution of larger brains and more sophisticated behaviors. The evolution of primate intelligence was a gradual process that unfolded over millions of years. Early primates had larger brains relative to their body size than most other mammals, but they were still relatively simple compared to modern apes and humans. Over time, however, primate brains continued to expand, particularly in areas associated with problem-solving, memory, and social cognition. The Old World monkeys, which split from the New World monkeys about 30 million years ago, evolved even more sophisticated cognitive abilities. They developed complex communication systems, used tools in some species, and showed evidence of cultural transmission, the ability to learn behaviors from other group members and pass them on to future generations. The apes, which evolved from Old World monkey ancestors about 25 million years ago, represented another major leap in cognitive evolution. They had even larger brains, more sophisticated problem-solving abilities, and more complex social structures. The great apes, orangutans, gorillas, chimpanzees, and bonobos all show evidence of self-awareness, tool use, and cultural transmission. About 7 million years ago, the lineage that would eventually give rise to humans split from the lineage that led to modern chimpanzees and bonobos. This split occurred in Africa, probably as a result of climate changes that fragmented the forest habitats where early apes lived. Some populations became isolated in shrinking forest patches, while others adapted to more open, savanna-like environments. The early hominids that adapted to savanna life faced new challenges that drove further cognitive evolution. The open grasslands provided less food and shelter than the forests, requiring more sophisticated foraging strategies and cooperation. The need to travel long distances between food sources favoured the evolution of bipedalism, the ability to walk upright on two legs. Bipedalism was a revolutionary adaptation that freed the hands for tool use and carrying. It also changed the anatomy of the skull and spine in ways that allowed for larger brains. As early hominids became more skilled at tool use and food processing, they could access higher quality foods that could support the metabolic demands of larger brains. The evolution of language was another crucial development in human cognitive evolution. The ability to communicate complex ideas through symbolic language allowed for unprecedented levels of cooperation and cultural transmission. Knowledge and innovations could be shared not just within groups, but across generations and even between distant populations. The mechanism of evolution, natural selection in detail throughout this entire magnificent story, from the first self-replicating molecules to the emergence of human consciousness. One fundamental process has driven the diversity and complexity of life: natural selection. This elegant mechanism, first clearly articulated by Charles Darwin in 1859, is both simple in concept and profound in its implications. Imagine natural selection as a gentle but persistent gardener, constantly tending to the garden of life. This gardener doesn't create new plants or decide which ones should grow, but rather creates conditions that favour certain traits over others. The gardener doesn't work quickly, changes occur over many generations, but the cumulative effect is profound. Natural selection operates through a simple but powerful process that requires just three conditions: variation, inheritance, and differential reproductive success. In any population of organisms, individuals vary in their traits. Some may be faster runners, others may have better camouflage, still others may be more resistant to disease. This variation is the raw material on which natural selection acts. For natural selection to work, these variations must be heritable. They must be passed from parents to offspring. In the molecular world, this inheritance occurs through the copying of DNA, RNA, or other genetic material. The information that specifies an organism's traits is encoded in its genes and transmitted to its descendants. The third requirement is differential reproductive success. Individuals with certain traits must have more offspring than individuals with other traits. This doesn't necessarily mean that the fittest individuals are the strongest or most aggressive. Fitness, in evolutionary terms, is measured solely by reproductive success. An individual that survives to reproduce and raises many offspring to maturity is more fit than an individual that fails to reproduce, regardless of other qualities. When these three conditions are met, natural selection inevitably follows. Individuals with traits that enhance their survival and reproduction will leave more offspring than those with traits that reduce their reproductive success. Over time, beneficial traits become more common in the population, while harmful traits become less common. This process can be illustrated with countless examples from the natural world. Consider the peppered moths of industrial England. Before the Industrial Revolution, most peppered moths were light-coloured, which provided excellent camouflage against the pale bark of trees. Dark-coloured moths were rare because they were easily spotted and eaten by birds. During the Industrial Revolution, pollution darkened the tree trunks in industrial areas. Suddenly, the light-coloured moths became conspicuous against the dark bark, while the previously rare dark-coloured moths were well camouflaged. The frequency of dark-coloured moths increased dramatically in polluted areas, while light-coloured moths remained common in unpolluted areas. This example illustrates several important principles of natural selection. First, the environment plays a crucial role in determining which traits are advantageous. Second, natural selection doesn't create new traits. It acts on existing variation in the population. Third, the process can operate relatively quickly when selective pressures are strong. Natural selection can take several different forms. Directional selection favours individuals at one extreme of a trait distribution. For example, if larger size is advantageous, the average size of individuals in a population will increase over time. Stabilising selection favours intermediate values of a trait, eliminating individuals at both extremes. Disruptive selection favours individuals at both extremes while selecting against intermediate values. Sexual selection is a special form of natural selection that operates on traits related to mating success. These traits may not enhance survival and may even reduce it, but they increase an individual's ability to attract mates or compete with rivals. The elaborate plumage of peacocks, the antlers of deer, and the complex songs of birds all evolve through sexual selection. Natural selection can also operate at different levels of biological organization. While we typically think of selection acting on individual organisms, It can also act on genes, cells, groups, or even species. Gene-level selection helps explain the evolution of selfish genetic elements that enhance their own transmission even at the expense of their host organisms. Group selection, while controversial, may help explain the evolution of cooperation and altruism. If groups with more cooperative individuals are more successful than groups with more selfish individuals, then cooperative traits may spread even if they reduce individual fitness within groups. The power of natural selection lies in its ability to create the appearance of design without a designer. Through the accumulation of small, favourable changes over many generations, natural selection can produce structures and behaviours of extraordinary complexity and sophistication. The eye, often cited as an example of irreducible complexity, actually evolved through a series of intermediate stages, each of which provided some advantage to its possessor. The process began with simple, light-sensitive cells that could detect the presence or absence of light. These cells were useful for distinguishing day from night or for detecting the shadows of approaching predators. Over time, these cells became organized into patches, then into cup-shaped structures that could detect the direction of light. The addition of a lens allowed for image formation, while the development of muscles allowed for focusing and movement. Each step in this process was gradual, and each intermediate form was functional. There was no sudden leap from simple light detection to complex vision. Instead, there was a smooth continuum of increasingly sophisticated visual systems, each building on the innovations of the previous generation. Natural selection is not a conscious process. It has no goals, no foresight, and no intentions. It simply describes the inevitable result of differential reproductive success in populations of reproducing organisms. Yet the cumulative effect of this unconscious process has been the evolution of life forms of breathtaking beauty and complexity, the molecular basis of evolution. While Darwin understood the basic mechanism of natural selection, he couldn't explain how traits were inherited or how new variations arose. The molecular basis of evolution wasn't understood until the discovery of DNA and the development of modern genetics in the 20th century. We now know that the information needed to build and maintain an organism is encoded in its DNA, a long molecule composed of 4 different nucleotides: adenine, thymine, guanine, and cytosine. These nucleotides are arranged in specific sequences that spell out the instructions for making proteins, the molecular machines that carry out most cellular functions. The process of evolution operates at the molecular level through changes in DNA sequences. These changes, called mutations, can occur through several mechanisms. Sometimes DNA polymerase, the enzyme that copies DNA during cell division, makes mistakes, inserting the wrong nucleotide at a particular position. Other times, chemical or physical agents can damage DNA, causing changes in its sequence. Most mutations are neutral. They don't significantly affect the organism's survival or reproduction. Some mutations are harmful, reducing the organism's fitness. But occasionally, a mutation is beneficial, providing some advantage to its carrier. These beneficial mutations are the raw material for evolutionary change. The genetic code is redundant, meaning that multiple DNA sequences can code for the same protein. This redundancy provides a buffer against harmful mutations and creates opportunities for evolutionary change. A mutation that doesn't change the protein sequence, a synonymous mutation, can still affect the organism by changing how much protein is produced or when it's produced. Gene regulation, the control of when and where genes are expressed, is another important source of evolutionary variation. Small changes in regulatory sequences can have large effects on development and adult characteristics. Many of the differences between closely related species appear to result from changes in gene regulation rather than changes in protein sequences. The discovery of DNA also revealed the molecular basis of heredity. Genes are discrete units of hereditary information, and they are passed from parents to offspring according to predictable patterns. Mendel's laws of inheritance, discovered in the 19th century through experiments with pea plants, make perfect sense in light of our understanding of DNA and chromosomes. Sexual reproduction shuffles genetic material in ways that create new combinations of traits in each generation. During meiosis, the process that produces gametes, eggs and sperm, homologous chromosomes pair up and exchange segments in a process called crossing over. This recombination creates new combinations of alleles, different versions of genes that have never existed before. The molecular clock is another important concept in evolutionary biology. Because mutations occur at roughly constant rates, the amount of genetic difference between two species can be used to estimate when they diverged from a common ancestor. This molecular approach has revolutionized our understanding of evolutionary relationships and has allowed us to construct detailed evolutionary trees for many groups of organisms. The unity of life, evidence for common descent. Despite the incredible diversity that evolution has produced, all life shares fundamental similarities that reveal our common ancestry. These similarities exist at every level of biological organization, from the molecular to the anatomical, and they provide compelling evidence for the unity of life. At the molecular level, all living things use the same genetic code. The sequence of nucleotides in DNA determines the sequence of amino acids in proteins, and this relationship is nearly universal across all forms of life. The fact that a human gene can function in a bacterial cell, or that a bacterial gene can function in a human cell, demonstrates the fundamental unity of life at the molecular level. All cells use the same basic biochemical pathways for energy metabolism. The process of glycolysis, which breaks down glucose to produce energy, is virtually identical in bacteria, plants, and animals. The citric acid cycle, which further processes the products of glycolysis, is also highly conserved across different forms of life. These similarities are not coincidental. They reflect our shared evolutionary heritage. The genetic code itself provides powerful evidence for common descent. There are many possible ways to assign the 64 possible 3-nucleotide combinations codons to the 20 amino acids used in proteins. The fact that virtually all life uses the same genetic code suggests that all modern organisms inherited this code from a common ancestor. Even the structure of cells reveals our common ancestry. All cells are surrounded by membranes made of phospholipids, all use ribosomes to synthesize proteins, and all store their genetic information in nucleic acids. These fundamental similarities suggest that all life descended from a common ancestor that had already evolved these basic cellular features. Anatomical similarities also provide evidence for common descent. The forelimbs of mammals, for example, all share the same basic bone structure, despite being adapted for very different functions. The wing of a bat, the flipper of a whale, the leg of a horse, and the arm of a human all contain the same bones arranged in the same pattern. This similarity is best explained by common descent from an ancestor that had this particular limb structure. Embryological evidence also supports common descent. The early embryos of vertebrates are remarkably similar, even though the adult forms are very different. Fish, amphibians, reptiles, birds, and mammals all develop pharyngeal arches during embryonic development, even though these structures form gills in fish but contribute to the jaw and ear structures in mammals. The fossil record provides another line of evidence for common descent. Fossils show a progression from simple to complex forms over time, and they reveal transitional forms that link major groups of organisms. The evolution of whales from land mammals, for example, is documented by a series of fossils that show the gradual transition from four-legged land animals to fully aquatic whales. Biogeographical patterns also support common descent. The distribution of species around the world makes sense in light of evolutionary history and continental drift. Islands that have never been connected to continents have unique faunas that evolved from the few species that were able to colonize them. Continental areas that were once connected share similar species, even if they are now separated by oceans. The dance of coevolution. Evolution is not just about individual species changing over time, it's also about the intricate relationships between species and how they evolve in response to each other. This process, called coevolution, has produced some of the most beautiful and complex patterns in nature. The relationship between flowering plants and their pollinators is one of the most striking examples of coevolution. About 130 million years ago, the first flowering plants appeared, and they quickly entered into partnerships with insects and other animals that could carry their pollen from flower to flower. This partnership was mutually beneficial. The plants received reproductive services, while the animals received nectar and pollen as food. Over millions of years, plants and their pollinators have co-evolved in remarkable ways. As flowers evolved bright colours and sweet fragrances to attract pollinators, insects evolved better colour vision and more sensitive chemical receptors to locate flowers. As flowers evolved deep, narrow tubes to hold their nectar, some insects evolved longer tongues to reach it. As flowers evolved different shapes and sizes, different groups of pollinators specialized on different types of flowers. This coevolutionary process has led to incredible diversity in both flowering plants and their pollinators. There are now more than 300,000 species of flowering plants and hundreds of thousands of species of pollinating insects. Many of these relationships are highly specialized, with particular plant species being pollinated by only one or a few species of insects. The relationship between plants and herbivores provides another example of coevolution. As plants evolved chemical defenses to deter herbivores, herbivores evolved ways to overcome these defenses. Some insects evolved enzymes that could break down plant toxins, while others evolved behavioral strategies to avoid the most toxic parts of plants. This evolutionary arms race has led to the evolution of thousands of different plant defensive compounds and the corresponding detoxification mechanisms in herbivores. The diversity of alkaloids, terpenes, and other secondary metabolites in plants is largely a result of this coevolutionary process. Predator-prey relationships also show evidence of coevolution. As prey animals evolved better camouflage, predators evolved keener eyesight. As prey evolved faster running speeds, predators evolved more efficient hunting strategies. As prey evolved protective armor, predators evolved stronger jaws and sharper teeth. The cheetah and the gazelle provide a classic example of this predator-prey coevolution. Cheetahs are the fastest land animals, capable of reaching speeds of 70 miles per hour. But gazelles are also extremely fast and agile, and they have evolved sophisticated escape behaviors that allow them to avoid capture. This ongoing evolutionary arms race has pushed both species to remarkable levels of athletic performance. Coevolution also occurs between species that have mutualistic relationships. The relationship between flowering plants and their pollinators is one example, but there are many others. Cleaner fish and their clients have co-evolved elaborate recognition signals and behaviors that allow them to identify each other and carry out their cleaning services safely. The relationship between ants and acacia trees provides another example of mutualistic coevolution. Some acacia trees provide ants with food and shelter in exchange for protection from herbivores. The ants attack any animal that tries to eat the acacia leaves, and they also remove competing vegetation from around the tree. This relationship has led to specialized structures in both the ants and the trees that facilitate their partnership. Even within organisms, coevolution occurs between different parts of the genome. Genes that work together to produce a particular trait often evolve in coordinated ways. The evolution of color vision, for example, required coordinated changes in the genes that produce the light-sensitive pigments in the eye and the genes that process visual information in the brain. The evolution of complexity. One of the most remarkable aspects of evolution is its ability to produce increasingly complex structures and behaviors over time. From the simple self-replicating molecules of the early Earth to the sophisticated organ systems of modern animals, evolution has consistently generated new levels of organization and complexity. But how does complexity evolve? At first glance, it might seem that natural selection should favor simplicity over complexity. Simple organisms require fewer resources to build and maintain, and they have fewer components that can malfunction. Yet the history of life shows a clear trend toward increasing complexity. The key to understanding the evolution of complexity lies in recognizing that complexity can be advantageous if it provides new capabilities or improves existing ones. The evolution of multicellularity, for example, allowed organisms to grow larger, develop specialized tissues, and exploit new ecological niches. The added complexity was more than compensated for by the advantages it provided. The evolution of complexity often proceeds through a process called duplication and divergence. When a gene or an organ system is duplicated, one copy can continue to perform the original function while the other copy is free to evolve new functions. This process has been crucial for the evolution of many complex systems. The evolution of the vertebrate immune system provides a good example of this process. The adaptive immune system, which can recognize and remember specific pathogens, evolved from a simpler innate immune system through a series of gene duplications and modifications. The duplicated genes evolved new functions that allowed for more sophisticated immune responses. The evolution of developmental complexity has been particularly important for the evolution of animal body plans. The Hox genes, which control the development of body segments and appendages, evolved through a series of duplications from a simple ancestral gene. As these genes diversified, They gained the ability to specify different body regions and appendages, allowing for the evolution of increasingly complex body plans. The evolution of nervous systems provides another example of increasing complexity. The first nervous systems were simple networks of nerve cells that could coordinate basic responses to stimuli. Over time, these networks became more centralized and hierarchical, with specialized regions for processing different types of information. The evolution of brains represents the pinnacle of nervous system complexity. Brains are incredibly complex organs that can integrate information from multiple sensory modalities, store memories, make decisions, and control complex behaviors. The human brain, with its 86 billion neurons and trillions of synaptic connections, is one of the most complex structures in the known universe. Yet even this complexity evolved through natural selection operating on simpler precursors. The basic building blocks of nervous systems, neurons and synapses, are found in even the simplest animals. The evolution of brain complexity involved the elaboration and reorganization of these basic components, not the creation of entirely new ones. The evolutionary history of Earth's climate. The evolution of life and the evolution of Earth's climate have been intimately connected throughout the planet's history. Organisms have not only adapted to climate changes, but have also played crucial roles in shaping the climate itself. The early Earth had a very different atmosphere than today. There was little or no oxygen, and the atmosphere was dominated by carbon dioxide, methane, and other gases that would be toxic to most modern organisms. The evolution of photosynthesis by cyanobacteria fundamentally changed the atmosphere, gradually increasing oxygen levels and reducing carbon dioxide concentrations. This change in atmospheric composition had profound effects on the evolution of life. The increase in oxygen levels allowed for the evolution of aerobic respiration, which is much more efficient than anaerobic metabolism. The reduction in carbon dioxide levels helped moderate global temperatures and may have contributed to the evolution of more complex life forms. The evolution of land plants had another major impact on Earth's climate. Plants remove carbon dioxide from the atmosphere through photosynthesis and store it in their tissues. When plants die and are buried, this carbon can be sequestered for long periods, effectively removing it from the atmosphere. The evolution of forests and other terrestrial ecosystems has played a crucial role in regulating atmospheric carbon dioxide levels. The evolution of marine organisms has also influenced climate through their effects on ocean chemistry. Many marine organisms, from microscopic plankton to large mollusks, build shells and skeletons out of calcium carbonate. When these organisms die, their shells can accumulate on the ocean floor, forming limestone deposits that lock up large amounts of carbon. Throughout Earth's history, there have been several major climate events that have had profound effects on the evolution of life. The snowball Earth episodes, when the planet was almost entirely covered by ice, may have spurred the evolution of multicellular life. The warm climate of the Mesozoic era allowed for the evolution and diversification of dinosaurs. The cooling climate of the Cenozoic era favored the evolution of mammals and flowering plants. The current era of human-induced climate change represents a new chapter in the relationship between life and climate. For the first time in Earth's history, a single species is having a major impact on global climate through its activities. The rapid increase in atmospheric carbon dioxide levels due to fossil fuel burning is causing global temperatures to rise at an unprecedented rate. This climate change is already having effects on the evolution of life. Some species are adapting to warmer temperatures through genetic changes, while others are shifting their ranges toward the poles or to higher elevations. The timing of seasonal events like flowering and migration is changing in response to shifting temperature and precipitation patterns. The evolution of symbiosis. One of the most important processes in evolution has been the formation of symbiotic relationships between different species. Symbiosis, the close and long-term interaction between different species, has been a major source of evolutionary innovation and has led to some of the most important transitions in the history of life. The evolution of eukaryotic cells through endosymbiosis is perhaps the most famous example of symbiosis in evolution. The engulfment of bacterial cells by larger host cells led to the evolution of mitochondria and chloroplasts, organelles that are essential for aerobic respiration and photosynthesis. This symbiotic relationship was so successful that it has been maintained for over 1 billion years. But endosymbiosis is just one form of symbiosis. There are many other types of symbiotic relationships that have played important roles in evolution. Mutualistic relationships where both partners benefit are particularly common and have led to some remarkable evolutionary innovations. The relationship between legume plants and nitrogen-fixing bacteria provides a classic example of mutualistic symbiosis. Legumes provide the bacteria with carbohydrates and a protected environment in specialized root nodules. In return, the bacteria convert atmospheric nitrogen into ammonia, which the plant can use to synthesize amino acids and other nitrogen-containing compounds. This symbiotic relationship has allowed legumes to colonize nitrogen-poor soils where other plants cannot survive. It has also had important ecological effects, increasing the availability of nitrogen in ecosystems and supporting the growth of other plants. The evolution of this symbiosis has been crucial for the success of legumes, which now include many important crop plants like beans, peas, and soybeans. The evolution of mycorrhizal relationships between fungi and plant roots is another important example of mutualistic symbiosis. The fungi help plants absorb water and nutrients from the soil, while the plants provide the fungi with carbohydrates. This relationship is so ancient and widespread that it may have been essential for the colonization of land by plants. Lichens represent another remarkable example of symbiosis. These organisms are actually composite entities consisting of a fungus and a photosynthetic partner, either an alga or a cyanobacterium. The fungus provides structure and protection, while the photosynthetic partner provides energy through photosynthesis. This partnership has allowed lichens to colonize some of the most extreme environments on Earth, from Arctic tundra to bare rock surfaces. The evolution of symbiotic relationships between animals and microorganisms has also been important for animal evolution. The digestive systems of many herbivorous animals contain symbiotic bacteria that help break down cellulose and other plant materials that the animals cannot digest on their own. This symbiosis has allowed animals to exploit plant resources that would otherwise be unavailable to them. The human microbiome provides a fascinating example of how symbiotic relationships continue to evolve. The trillions of bacteria that live in and on our bodies play crucial roles in digestion, immune function, and even behavior. These relationships are so important that some scientists consider humans to be superorganisms consisting of human cells and their microbial partners. The evolution of behavior. The evolution of behavior is one of the most fascinating aspects of evolutionary biology. Unlike physical structures, behaviors don't fossilize, so we must infer their evolutionary history from studies of modern organisms and their genes. The capacity for behavior is ultimately based on the nervous system, so the evolution of behavior is closely linked to the evolution of neural complexity. Even the simplest animals show basic behaviors like moving toward food or away from danger. As nervous systems became more complex, behaviors became more sophisticated. The evolution of social behavior has been particularly important for many species. Living in groups provides many advantages, including protection from predators, improved foraging efficiency, and cooperative care of young. But group living also creates new challenges, including competition for resources and the potential for conflicts between group members. The evolution of cooperation has been a major puzzle in evolutionary biology. At first glance, it seems that natural selection should favor selfish behavior over cooperation. Why would an individual sacrifice its own interests to help others? The answer lies in understanding the genetic basis of behavior and the conditions under which cooperative behavior can evolve. Kin selection provides one explanation for the evolution of cooperation. If an individual helps its relatives, it is indirectly helping copies of its own genes that are carried by those relatives. This can make altruistic behavior advantageous from a genetic perspective, even if it reduces the individual's own survival or reproduction. The evolution of eusociality in insects provides a striking example of kin selection in action. In eusocial species like ants, bees, and termites, most individuals are sterile workers that devote their lives to helping their relatives reproduce. This extreme form of cooperation is made possible by the unusual genetic system of these insects, which creates high levels of genetic relatedness between sisters. Reciprocal altruism provides another mechanism for the evolution of cooperation. If individuals can recognize each other and remember past interactions, they can develop strategies of conditional cooperation where they help others who have helped them in the past. This tit-for-tat strategy can be evolutionarily stable if the benefits of cooperation outweigh the costs. The evolution of communication has been crucial for the development of complex social behaviors. Animals have evolved a remarkable diversity of communication systems, from the chemical signals of bacteria to the complex vocalizations of birds and mammals. These communication systems allow individuals to coordinate their behavior, share information, and maintain social relationships. The evolution of learning and memory has also been important for behavioral evolution. The ability to learn from experience allows animals to modify their behavior based on changing environmental conditions. This flexibility can provide significant advantages in unpredictable environments. The evolution of culture represents another major transition in behavioral evolution. Culture is the transmission of information and behaviors through social learning rather than genetic inheritance. This allows for much more rapid adaptation to changing conditions than would be possible through genetic evolution alone. Human culture represents the most extreme example of cultural evolution. Our ability to accumulate knowledge and innovations over generations has allowed us to adapt to a wide range of environments and to develop technologies that far exceed what any individual could create alone. This cultural evolution has accelerated dramatically in recent centuries, leading to rapid changes in human societies and lifestyles. The future of evolution. Evolution didn't stop with the appearance of humans. It continues today, as it has for billions of years. All around us, species are still adapting to changing environments, still evolving new traits, still experimenting with new forms of life. We can observe evolution in action in many different contexts, from the development of antibiotic resistance in bacteria to the adaptation of urban animals to city life. The rapid pace of environmental change in the modern world is creating new selective pressures that are driving evolutionary change. Climate change is already causing species to shift their ranges and modify their behaviors. Pollution is creating new chemical environments that favor organisms with novel detoxification mechanisms. Habitat fragmentation is creating small, isolated populations that may evolve in new directions. Human activities are also directly influencing the evolution of other species. The use of antibiotics has led to the evolution of drug-resistant bacteria, while the use of pesticides has led to the evolution of resistant insects. Hunting and fishing pressure can lead to evolutionary changes in the size and behavior of target species. The evolution of human populations is also continuing, though it may be difficult to observe over short timescales. Changes in diet, lifestyle, and disease exposure are creating new selective pressures that may influence human evolution. The ability to treat genetic diseases and other medical conditions may also be altering the course of human evolution. One of the most exciting frontiers in evolutionary biology is the study of evolutionary developmental biology, or EVO-DEVO. This field examines how changes in developmental processes can lead to evolutionary change. By understanding how genes control development, we can better predict how organisms might evolve in response to changing conditions. The development of new technologies is also opening up new possibilities for understanding and even directing evolution. Genetic engineering techniques like CRISPR-Cas9 allow scientists to make precise changes to DNA sequences, potentially accelerating the pace of evolutionary change. While these technologies raise important ethical questions, they also offer the possibility of addressing major challenges like disease and environmental degradation. As we look to the future, we can only imagine what new forms evolution might produce. Will intelligence like ours evolve again in other species? Will life find ways to survive and thrive in the face of climate change and other environmental challenges? Will evolution take life beyond Earth to other planets and solar systems? One thing is certain, as long as there is life, there will be evolution. The same fundamental processes that created the first cells will continue to operate generation after generation, creating new forms of beauty and complexity that we can barely imagine. The story of evolution is far from over. In fact, it may be just beginning. The eternal cycle and our place in the story, and so we return to where we began, in the warm, shallow pools of the early Earth, where the first organic molecules began their ancient dance. The same carbon atoms that were part of those first compounds may now be part of your DNA, part of a leaf on a tree outside your window, part of a bacterium in the soil beneath your feet. This is the eternal cycle of life, matter constantly rearranging itself into new forms, new patterns, new possibilities. The atoms in your body have been recycled countless times throughout Earth's history. They may have been part of a dinosaur, a flowering plant, marine bacterium, or a star that exploded billions of years ago. You are literally made of stardust, connected to the cosmos through the endless recycling of matter and energy. We are not separate from the process of evolution. We are part of it, temporary arrangements of atoms that have been borrowed from the universe and will one day return to it. The oxygen you breathe was first produced by cyanobacteria billions of years ago. The cells in your body carry the descendants of ancient bacterial symbionts. The very ability to think and dream evolved over millions of years of gradual change. Your existence is the result of an unbroken chain of successful reproduction stretching back to the very origin of life. Every one of your ancestors, from the first self-replicating molecules to your immediate parents, survived long enough to reproduce. You are the current endpoint of a lineage that has persisted for nearly 4 billion years. This perspective can be both humbling and inspiring. We are just one small part of a vast web of life that connects all living things. We share a common ancestry with every other organism on Earth. From the smallest bacterium to the largest whale, we are all leaves on the same vast tree of life. Yet we are also unique. The evolution of human consciousness has created a new kind of awareness in the universe, the ability to understand and appreciate the very process that created us. We can contemplate our origins, study our evolutionary history, and even influence the future course of evolution. With this knowledge comes responsibility. We are the first species in Earth's history to have the power to dramatically influence the evolution of life on a global scale. Our actions are already affecting the survival and evolution of countless other species. We have the opportunity to be wise stewards of the evolutionary process, or to disrupt it in ways that could have catastrophic consequences. As you prepare for sleep, remember that you are part of this magnificent story. The processes that created you are still operating in your body. Your cells are dividing, your immune system is adapting to new challenges, your nervous system is processing information and forming memories. Even in sleep, you are participating in the ancient dance of life. The story of evolution is not just about the past, it's about the present and the future as well. Every day, evolution continues to unfold in countless ways, from the molecular level to the ecosystem level. New mutations arise, new species evolve, new ecological relationships form. The story that began with simple molecules in primordial seas continues to unfold in ways we can barely imagine. In your dreams tonight, perhaps you'll sense the deep time that flows through your veins, the ancient rhythms of change and adaptation that have brought you to this moment. You are evolution's gift to the universe, a conscious being capable of understanding and appreciating the very process that created you. The ancient dance continues even as you sleep, in the gentle processes of cellular repair, in the quiet workings of your immune system, in the dreams that flicker through your mind like ancient memories. You are part of the eternal cycle, connected to all life through the invisible threads of evolution. Rest now, child of the cosmos, descendant of the first cells, inheritor of 4 billion years of evolutionary innovation. The story continues, and you are both its product and its author. In the morning, you awake as a new chapter in the endless book of life, ready to contribute your own verse to the magnificent poem of existence. Sleep well and dream of deep time, where molecules dance and species bloom, where intelligence emerges from simplicity, and where the future unfolds like a flower opening to the sun. You are part of the greatest story ever told, the story of life itself, unfolding across the vast expanse of time, creating beauty and complexity and wonder at every turn. Sleep peacefully, knowing that you are both a product of evolution and a participant in its continuing story. The ancient dance goes on, and you are part of its eternal rhythm.